Hi everybody, my name is Leonard Carsten from Bielefeld University. I'm here to guide you through my poster with the subject Generation of Antibody-Drug Conjugates using the formal glycine-generating enzyme. The formal glycine-generating enzyme, or in short FGE, specifically oxidizes its cysteine within the consensus sequence CXPXR to the non-canonical amino acid C-alpha-formylglycine, also called F-gly. This recognition motif, or aldehyde tag, can be genetically inserted into proteins to perform a site-specific F-gly conversion. With the aldehyde moiety of the F-gly, the protein becomes addressable for bioconjugation chemistry to generate antibody-drug conjugates. Therefore, we designed various antibody constructs for a specific targeting of the epidermal growth factor receptor. The EGF receptor is overexpressed in many cancer types and validated target. We chose the single-chain variable fragment 425 and the designed ankyrin repeat protein DARPin E01, both directed against the EGF receptor, and fused them to the Fc domain of the human IgG1 antibody. These antibody constructs are C-terminally equipped with aldehyde tags to introduce the F-gly. The electrophilic aldehyde moiety of F-gly can be addressed selectively by strong nucleophiles. We established a bioconjugation chemistry with modified tryptamines or N-aliphatic substituted pyrrosolones, which are used in the Hipps ligation or the trapped and tandem-Knoevenagel condensation reactions. These inexpensive building blocks can be combined with highly efficient strain-promoted acid alkyne cycloadditions for selective mono or double derivatization with DBCO modified payloads. This two-step synthesis greatly reduces the need of expensive drug-containing payloads to stoichiometric amounts. We also combined prokaryotic and eukaryotic F-gly generating systems with varying specificity for different recognition motifs. The first F-gly gets introduced by the human FGE with its specificity for the CTPSR motif. The empty FGE from Mycobacterium tuberculosis attains a substrate promiscuity for the proline-free motif CTAGR after kappa reconstitution or in presence of kappa sulfate. This sequentially introduction of two F-gly residues allowed the generation of a carboxyfluorescein labeled ADC containing a monomethyl auristatin payload. We performed live cell imaging experiments with our dye-labeled antibody constructs on A431 cells with a high level of EGF receptors and MCF7 cells with a notably low level of EGF receptors present on the cell surface. Both constructs internalized and co-localized with the lysosomes upon binding to EGF receptor-rich A431 cells. Our isolated single-chain Fc MMAE conjugate revealed a drug-to-antibody ratio of 1.3 to 1.7. We performed in vitro cytotoxicity assays with our MMAE ADC on the same tumor cell lines. The ADC shows a specific effect against EGFR-presenting cells with an IC50 value of 0.05 nanomolar against A431 cells. Thank you for your attention. Feel free to visit my poster room for any questions or if you'd like to start a discussion. See you.